time. So hi everyone, my name is Karen. I am on the DevRel team at Databricks, and we're happy to be able to bring this online meetup to you so quickly. And thank you all for joining. And we all hope that you and your families are safe and healthy. And we hope you enjoy this session. So I'm going to pass it along to my colleague Denny Lee. He's also on the DevRel team. He's a developer advocate. So he's going to kick things off and get it started here.
So hi there. Again, thanks very much, Karen, for the introduction. My name is Denny Lee. I'm a developer advocate here at Databricks. But before that, I was actually a pseudo biostatistician. I was actually doing a master's in biostats at University of Washington. I actually was working with the on the ADRIP project, which was working on HIV and AIDS research with the Fred Hutch Cancer Research Center and University of Washington Virology Lab here in the Pacific Northwest. So the reason we decided to shift gears from our original session today that was about addressing GDPR and CCPA was because due to the current health crisis, health concerns, we thought this might be a very good timing for us to go ahead and talk about a session like this. Even though I never completed my biostats degree, just as a quick call out, the reason why I didn't is because Microsoft offered me money to go work versus me paying for a degree. So yeah, I took the money. But nevertheless, I do actually have a master's in biomedical informatics, and I do have a background in medicine. I actually have a degree in physiology. That does not make me an expert. As much as my parents wanted me to be a doctor, Asian parents, no, I'm not. Okay, so let's not bother with the pretense here. But I do have many friends and many colleagues here in the Seattle area that are currently helping to fight the coronavirus and helping all of our patients. So before we go into the data science side of things, I want to be very clear. The number one thing you can do to help everybody is wash your hands, okay? I cannot overemphasize that enough. The number two thing you can do is social isolation. This is the reason why I'm currently sitting in my laundry room as opposed to being out and about, because it's the right thing to do, okay? So those are the two most important things. For those who want to see if they can possibly help, we put a panel of really cool data scientists together today to talk about the different data sets we have. There's actually probably more, by the way, but the ones that we happen to be working with: the South Korean data set out of Kaggle, the COVID-19 data set that's also on Kaggle, and the Johns Hopkins data set that's actually sitting in GitHub. Okay, and we're going to be showcasing these notebooks. We're going to be saving them. We're probably going to be writing this on Databricks right now, but we're actually going to be saving these as iPython notebooks so you can run them locally on your Jupyter instance. So what will happen is that after this session is done, we're going to prop on both the Spark Online Meetup as well as YouTube, where this video is playing as well, links to those notebooks that you can download and work with yourself with these public data sets. Okay, so what we're here is simply just trying to encourage you folks to go ahead and take your data science knowledge and see if you can possibly help. But obviously, understand that, still the number one thing you can do is wash your hands. The number two thing you can do is go ahead and social distancing. And if you have time, try to be on the lookout for your friends, for your elderly parents or your elderly colleagues. And on top of that, just like a lot of the folks here in the Pacific Northwest, and I'm sure it's happening everywhere right now, go ahead and donate to the various meal deliveries to your healthcare providers. Okay, so just because to help them out. Okay, these are the number 1, 2, and 3 things you should do, by the way. Okay, now, since we're at home and want to do a little bit of data science, this is actually also a fun thing to do to try to analyze the data. Okay, so saying that, I did want to first introduce Vinny. Vinny, you're going to introduce yourself, obviously, but I'd like to start with you to go ahead and actually present your session. Okay, so let's start with you, Vinny, and please take it away.
Thanks, Denny. Hi, guys. I am Vinny Jaiswal, Customer Success Engineer at Databricks. I have been with Databricks since late 2018, and I serve as a trusted advocate for our customers to make sure they are happy with our platform. I have been working with big data industry and in general, like 7 years. And I have master's degree in information technology and management from UT Dallas. And I also worked with Citibank and Southwest Airlines before. So that's a little bit about me. Today, I am going to just show some of the, all right, let me share my screen. So I'm going to share with you a quick notebook. I have analyzed South Korea dataset, and you can find this dataset, as Denny mentioned, later. This was available publicly, and I just started analyzing some quick points since since there is a lot of word about how the cases have been found, and I'm just going to walk you through some of the insights that I have seen in the data. So I'm using Databricks notebook here, and you can do the same thing in either Jupyter or other platforms as you wish. Here, if you can see, I have patient CSV and just writing some SQL codes to understand what the data is. So this is how my data set looks like. You can see that there is patient ID, gender, birth year, country, of course, Korea, but there have been cases from other countries which visited South Korea, and we have other details like confirmed date, release date, and things like that. So the first thing I want to show you is number of patients in each city. You can see that there have been rises in cases, and it started with the capital city which has the most number of patients, and then you can see other regions as well. I'm just running a simple SQL query to find the patient counts for each region. So we can see the trend here. Another insightful thing I noticed is how about we filter the cases based on infection reasons. So what I did is I calculated month over month what type of infection reason the patient was affected by. So as you can see, in January, a lot of cases were from visiting the Wuhan region, and then it eventually developed with the contact with existing infected people. So you can see that's where the trend started. In month of February, all these cases which you see in orange are contact with patient, and it just grew organically. Then people started visiting other cities, and second biggest infection reason was visit to Daegu, and it just eventually spiked up. So you can see like the most common trend is contact with other people. So that's why wash your hands, and we have all this lockdown in place. Other interesting thing about this data is confirmed count. So let's see like what kind of trend we have seen depending on date and number of patients. So it started in January. Slowly it picked up because of many reasons: travelers coming in contact with other patients, and then testing. A lot of people were not even aware that infection has happened to them. So it just started slowly trending. So this is the trend over a period of 3 months. Now let's talk about recovered and fatalities. So out of the cases that we are known about, these are the number of recovered patients, and I'm trying to analyze it based on like where the recovery is coming from. So from the graph, it looks like most patients are curable in the capital area region. So you can see that there are highest number of patients plus recoveries happening. Now let's talk about fatalities. What I did was I took the number of patients and their timelines from the confirmed date to the reported deaths, and just filtered by the region. And most of the fatalities which happened were from Daegu, which was the church event, and then Johnson book, and then the capital area. Now let's talk about percentage of recovered patient by infection reasons. So it looks like most patients which were in contact with other people got recovered. 40% of the total population of the reported confirmed cases were recovered. Second most recovery happened to the patients, which was 22% of total who visited to Wuhan. So these are some of the insights which can be derived from our data set. Number of fatality cases. So here you can see that the main fatalities came from church and the Denham hospital. And a lot of these cases which has unconfirmed death, maybe like for the reasons like they were not being tested, or they were not reported, or maybe they were not in a state where they can explain the reasons why the disease got to them. So these are some of the insights which we saw from the data set we had from South Korea. And a lot of it looks like the main takeaway from this is over a period of time, it just started happening, and it was mainly about coming to the contact with existing patients. Second takeaway is if we limit the contact, this is the cases can be controlled. So those are some of the things which we are also experiencing in our existing time frame. So that's about the analysis. Denny, you want to add anything?
No, this is great. Thanks very much. Actually, do me a small favor and scroll right to the bottom, so just we can show that call out, actually. Yes, percent of fatalities by infection reasons. So as you can tell, now remember, this is actually from Kaggle, the South Korean data set. Okay, and the South Korean data set actually is relatively complete because they actually have a very high rate of testing in South Korea. And so the fact that they can go ahead and specifically call out the various hospitals or the church, which was the main epicenter for South Korea, is pretty substantial. But the fact that they also cannot go ahead and actually explain the reasons, which is the vast majority of the group, is actually very telling, which is basically calling out that, yes, once it spreads, and we actually can't really do that network effect to figure out, is there a single cause anymore, because it has that network effect kicking in. And so this is a really awesome work by Vinny. She did it over a few days, I guess, which is pretty cool. So there are actually some quick questions here that I thought were pretty cool for you. Vinny, if you could go ahead and sort of answer these questions here. So Vinny, I guess the first question is, why did you, basically, as you were looking at the South Korean data set here, what led you to actually ask these particular questions? Like, why did you decide to write the queries that you did?
Yeah, so this was based on like the news channels. I have been looking at the reasons, like I talked to my friends and what are some of the common questions that have been asked or that people want to know about. So that led me decide on some of the questions. Also, the data set present, the schema I had in the data set led me to find out those questions so I can find very relevant answers.
Cool. All right. And then related to that, so then basically, you yourself as a domain analyst, or yourself as an expert with the background that you have, like basically, is that the reason why, for example, you decided to write this in SQL, or is there a reason why you chose to go ahead and write the queries the way you wrote them?
Yeah, it was easier to do in SQL. I could do it in Python as well. So I can also mix and match different languages within this notebook. But the reason I selected SQL is because a lot of people whom we will share this notebook with, they may be more comfortable with SQL, so they can do easy analysis on SQL as well.
Perfect. So just as a finishing note then, there's some other questions that have popped in the Q&A. I'll actually take care of them in a separate session here. So no, in separate session here, that is, okay. But as Vinny had called out, she's got this awesome notebook that she created. We're going to be providing this as an iPython notebook shortly. So that way you can run this locally against the Kaggle dataset yourself, okay? So just as a quick call out, we are working on getting those links up. And so right now we have them currently as Databricks notebooks, but we are going to, like I said, convert them over. In fact, the final session today, Drew was actually going to showcase the fact that he actually was ahead of the curve of all of us and had already taken care of that. Okay, so there you go. Oh, and there is an interesting question that I will take over. So actually, Vinny, you can stop. Presenting. I will present my screen now. Okay. All right. Assuming I learn how to present my screen. That's a different story altogether. All right.
Thanks, Denny.
Thank you very much, Vinny. Appreciate it. All right, cool. So there's a couple of questions. By the way, can you? I notice there's some questions popping up in the chat. Please put them in the Q&A, just because it's actually easier for us to go ahead and organize. We're not going to necessarily answer the questions right away because each person is actually trying to present their what they're doing first. Okay. So the first question I'm going to go answer is any tips for developers who are relatively new to data science? And the quick question is that everybody who, irrelevant of your experience, your experience could be from a data engineering, could be SQL, could be algorithms. You can come in from a different angle. And actually this is very apropos for this particular data set, because I'm actually going to show you from this data set, the COVID-19 data set, from more of a data engineering perspective. Okay. And Chen Ying right afterwards is going to show it much more from a data science perspective. They're both equally important, right? This will become very apparent very shortly, why they're both really important. But the call out is that I would just start with that. Now, if you're really, really new to data science, I've never worked with it before, I would actually suggest to go ahead and work with Pandas and scikit-learn. This is probably the easiest way to get on board. The scikit-learn library, the website actually has a lot of good stuff that gets you jump started to learn how to do the stuff really quickly. Pandas is relatively easy. You can install it right on your laptop. So that way you're not actually going ahead and trying to do something brand new. It's just use your own laptop, whether it's Windows or Mac, doesn't matter, Linux for that matter. And you can just download the data sets and start working with it, okay? All right, and then finally, there's another question which I'm going to go ahead and also answer live, which is for these data sets, how often being refreshed? Actually, there are different organizations that are refreshing them on our different timelines. So for example, the South Korean data set was updated at least twice in the last week. The current COVID-19 working books data set here that we're going to be referring to as part of the COVID-19 Open Research Dataset Challenge on Kaggle, that data set's actually been updated. Right now they're on version 3. It's my understanding that they're updating it on a weekly basis, though I'm sure they're updating as fast as they can. If you want to check out, by the way, there's actually a subreddit on COVID projects called that actually goes ahead and has various people who are trying to go ahead and analyze, organize, cleanse the data even faster. So I'm not going to point to any particular project just because there are many, whether it's actually data science or 3D printing, by the way. So there's actually a wide variety of them. But the call out basically is that there's actually a lot of projects out there as well. To take whatever challenge you want to go and do, because our attitude is like, go out, do some analysis, see if you can help out. But again, wash your hands first. Okay, now that I'm jumping off the soapbox, pun intended, let's go ahead and talk about the COVID-19 data set. So what's cool about this one of the COVID-19 data set, it's actually a combination with Allen Institute, Chan Zuckerberg Institute, Microsoft Research, Georgetown, National Institutes of Health, and the White House. So we've got a bunch of collaborators, as you can tell right away. The data set here is actually, it's actually primarily JSON files. Okay, it's broken down to the commercial use subset, non-commercial use subset, and preprints that have not been peer reviewed. These are all papers that are about the coronavirus. Okay, and so we're going to be using this particular notebook on Databricks. Okay, but this notebook's, like I said, free to share, and we're going to be also converting this into an iPython notebook as well for folks to go ahead and work with. Okay, so the beginning of this notebook, I'm actually going to go a little different. First, Vinny had showed you, here's what we can do with the analysis of it. Now I'm going to be a little bit more boring and show you how you're going to go ahead and try to read all of this JSON, okay? Because that's what this COVID-19 data set is. And then hopefully you guys won't fall asleep, because Chen-Yin will follow up and she will do something much more interesting with this data set, okay? Because I'm going to do the boring data engineering stuff first. All right, so if I look at the schema of this JSON, and this is just a JSON schema.txt that they included within the data sets. All 3 or 4 data sets that they have as part of COVID-19 are actually here. And you'll notice it says things like the paper ID, metadata, like as in what's the title, the authors of it, what the abstract is, the body text, the text of it, and also all the bibliography references. And there's actually many, many, many references, okay? And so it's pretty cool stuff to have, but how do you make sense of it? Okay, so what I did is I said, okay, I'm going to go ahead and actually take all the JSONs, which actually we actually did upload this. If you happen to be using Databricks, we did upload this already to Databricks datasets, so you can just use them right away. If you don't, like I said, you can just go to the Kaggle. I actually included a link in the notebook to Kaggle to download the dataset itself. It's not very big, it's 2 gigabytes. Okay, so it's not like this gigantic dataset yet. Okay, now, I'll explain in a second why I have these Parquet variables, but the context is that I want to go ahead and convert them from JSON to Parquet. Okay, so why did I want to do that? Well, the reason why is because in order for me to just have Spark, and I've got 4 nodes, by the way, for this for the Spark cluster, just to read the 9,000 commercial use subset of JSON files. Each JSON contains information of the paper, like I said, the author, the title, the body text, and the bibliography. It's a pretty long-winded JSON, as you can tell here, okay? And so I went ahead and read it just to go ahead and do the initial read, which isn't actually doing a real process, okay? Basically, it took me almost 6 minutes just to read those 9,000 on 4 nodes. Okay, so it's pretty tiring. The good thing about reading in Spark, though, is this, because I went ahead and actually just simply put all 9,000 JSON files in one directory, and it was actually able to infer the schema anyways. So that's why it took so much time, because it was actually able to look at all the JSON, figure out what the schema was, and just automatically create the schema for me on the fly. That's why you see what you see here. Okay. Important to note, these JSONs are multi-line, i.e. they contain character returns. So if you don't specify this, when you try to read the JSON files, what will end up happening is you'll see corrupted record. That's all you'll see. And just because that means is simply because Spark did not recognize or understand what these JSON files were. So it is what it is. There you go. So, but make it multi-line, then you should be good to go. All right. So as we said here, here's the count, and it tells you right away, okay, there's 9,000 files. So cool stuff. All right. All right. And also I just verified it by just simply doing it. Let me look at the path. So just doing the ls on the file system and just do a count. All right. Now here's where the fun part happens. Okay. The fun part basically happens here, where you're going ahead and saying, let me go ahead and get the number of partitions to 286. In order to be able to, like I said, to read it, it took initially about 5 minutes, sorry, almost 6 minutes. But then when I wanted to write it out, it actually took me almost 14 minutes. Okay, so what I did is, because I have 4 nodes, I repartitioned the data and saved it as a Parquet. So instead of reading the individual JSON files, I saved it as 4 Parquet files. In fact, if you look here, here are the 4 Parquet files that you're seeing. Okay, these are the 4 Parquet files. All right, so because I did repartition as opposed to coalesce, I've minimized the skew. In other words, if I wrote coalesce here instead of repartition, which because coalesce would have been faster, the repartitioning actually went ahead and made sure the file sizes were roughly the same. The coalesce actually, when I ran the coalesce the first time, actually one of the files was massively bigger and the other 3 files were smaller. What that meant is that when I was trying to do a query against the data, if I was distributing this across 4 nodes, one node would take on more of a hit versus the other. Now, let's roll back a second. Why did I do all this? Okay, so I'd already told you 9,000 files. It took me about 6 minutes to go ahead and run the queries just to read it and do a count. Okay, well, because I went ahead and did this as Parquet, you'll notice that now when I run the query, it actually finished in 3 seconds. Okay, and to read the files, and it took less than 2 seconds, 1.5 seconds to go ahead and do the count. All right, so this is the data engineering aspect. And so for all you folks that are actually doing the COVID-19 data set, again, this notebook is going to be made available to you so you can follow up. But hopefully this will help you charge up and go through your data faster, because you're going to be able to go ahead and convert the JSON into Parquet, and you're going to be able to go ahead and run your queries significantly faster. When I was running queries before on the JSON, each one took me 5 or 6 minutes. But because I went ahead and converted to Parquet, now they're happening in seconds. Okay, so from a data engineering and data science perspective, that's significantly faster. Okay, so hence the reason why we went through the process of doing this. Okay, and the next, which I'm not really going to dive too much into, is basically the same thing. We just simply went ahead and provided the same conversion of the non-commercial data set, same idea. It took longer before. When I saved it to Parquet, I went ahead and the query went down significantly faster, and also for the peer review data sets. All right. All right, so Noel asked a great question. What's the benefit of running to get the number of partitions? Okay, in this particular case, I wasn't really, there's no true benefit to getting, I just wanted to call out what the number was. In other words, that there were 280 partitions before. What it does tell me, though, is that basically because we shoved the data in, right, I actually have a lot of partitions. And if I was to save it out, remember the overall size, and this, admittedly enough, the commercial data set was the largest, but the overall data size is only about 2 gigabytes, right? So we're not talking massive scale here. We're just simply talking about, like, you know, a small set of files, right? But I have these small set of files that are spread across 283 partitions. So if I had saved in Spark, or for that matter, if I was doing this in Pandas, same problem, by the way. Okay, I was to save it, I would actually save it out as 283 Parquet files, right? And that's bad, right? Because then there's the overhead of trying to read all of those little Parquet files, even though there's actually not that much data in each one. Instead, because like I said, I actually chose to do it as 4 just because the cluster setup, the Rocinante here that I have, happens to have 4 nodes. So I've saved it as 4. You could also just as easily turn around and tell me, dude, it's a pretty small thing. This Parquet has snappy codec as well, so it does a little bit of compression. You could have just saved this as 1 file and be done for the day. And that's valid. And in fact, that's actually what I'm planning to do for those folks who are going to be doing Pandas. Because you can actually in Spark go read those files, save it as a save as a Parquet file, and provided you remove the commit and the metadata files, then Pandas can actually go read that file too. Okay. So basically that's the real quick call out. And finally, the other call out for Noel is that does not tell you where the files are splittable. Yes and no. In this case, basically, there were 9,000 files to begin with. So basically, the idea of partitions in memory, we're basically taking those 9,000 and Spark created 283 partitions in memory and basically shoved those 9,000 files into 283 partitions in memory. By definition, if I write those partitions out without actually organizing it, and if I was to actually specify partition, or if I even did it with a single partition, usually what ends up happening is I have I'll get something like 280, or at least a heck of a lot more files than the 4 that I specified. And that's the reason why I didn't want to do that, just because it's the data size is relatively small in the first place. Okay. All right. Perfect. And we did have an anonymous person ask, are we going to talk about Delta Lake? We are not. In a future session, if you guys are interested, because we actually we're thinking about diving deeper onto each and every one of those notebooks. So Vinny's notebook, this notebook, and Chingying's stuff, and also Drew's notebook that'll be coming very shortly. We actually are thinking about going ahead and diving deeper into those situations where we will be doing streaming and we will be updating the data sets. In those cases, I may go ahead and decide to go ahead and include things like Delta Lake, but right now I'm just focusing on what's the best way for every single data scientist to make use of the data. And there's a sizable chunk of folks here that I'm sure are going to go ahead and tell me, I'd rather do this in iPython notebooks, locally with Pandas. That's fine. We are not actually asking you to use one version or the other. That's why we're actually going to save the stuff, these notebooks in that way. So that way you can go ahead and run this as an iPython notebook in Pandas as well. Okay. All right. So I did want to call out some quick analysis of the data prior to showing the cool stuff. Okay. So right now, like I said, I went ahead and read the data and I said, okay, cool. Let me go ahead and read the Parquet file. So that's what's great also is because I save this Parquet, then any subsequent notebook that I have, I can just go ahead and say, hey, let me just go read those files, Parquet files, and bam, I've got myself a nice data frame and I'm back up and running. Okay. So the first few cells here are very similar to the previous cells, so there's nothing really new here, okay? I'm just, again, showing the schema. It's pretty complicated. All right, so how do I want to make sense of the data? So in this case, I'm going to just use Spark SQL to help me try to look at the data. And then I know from this schema, JSON schema text, that the important things that I want to look at, because in this case, I was just wanting to have a little map it out and see which geo has what papers. Okay, so I can look at the paper ID. That gives me the count of the papers. Metadata, which tells me things like, okay, the title, but more importantly, the authors and their affiliation and their location. Inside the affiliation location, the affiliation, excuse me, there's the location. That location basically tells you where the author is from. Okay, so let's go do that. I'm going to go ahead and do select paper ID, metadata title. So this is, for example, the evolution of poxvirus vaccines. And here is the paper information. And as you can tell, here are all the array of authors and all their affiliations. Okay, so this is the author, and some of them will indicate exactly where they're affiliated. There's the affiliation, for example, and the location, and we're going to say Spain right here. All right, perfect. All right. So this tells you a little bit about that information. So we're diving into the JSON, trying to make sense of it. So now I'm saying, okay, let me break that out. So in this case, I'm going to go do an explode, because what I care about specifically is just the author information. I don't care about all the other information. I just care about the author information. So let me explode that out. What that basically means is that, as you remember, as you can tell, there are many, many, many authors for this first paper. Okay. All right, so that's great. That's actually good information, okay? See, all these authors here. Instead of actually having one row for each author, instead of having one row that contains all of these different authors inside here, where there's 6 of them, I'm gonna have separate rows for each author. So that way I can actually understand the author location. All right, so that's what this column is. And so that's why I exploded it. So I can say, okay, author 1 for the same paper, the evolution of poxvirus vaccines, right? Here's the 6 different rows for that particular paper. They have the same paper ID, the same name. I don't really need the title, but it's easier to read for everybody. So that's why I kept it in here, okay? And I can see where the affiliation is, okay? And where the location is, okay? So like same idea, country of Spain. So relatively straightforward. Okay, perfect. So that means if I'm lucky, I should just be able, in order to be able to go ahead and map this out, I should be able to go ahead and say, let me go ahead and take the author's location, the country, and map it to paper ID, and then I can do a count, and I'm good to go. Except, okay, problem number 1. I don't know where there's multiple authors, right? So which one do I choose? So I'm actually going to simply choose the minimum geo, because in some authors there are actually multiple geos. So literally, I'm just going to take the minimum one. That's actually probably not the best one, by the way. In fact, I probably should have ran a rank query to get to do it by first author, second author, third author, and then chosen just the first author. And in fact, actually, if you're interested for a subsequent session, that's exactly what I'll do. Okay, but so just as a call out, I'm actually showing my mistakes. Okay, but for now, I'm just doing a min. So that's what I did here. Okay, I basically said, okay, give me the min country from this. All right, so this is perfect. So each paper ID, the minimum country, as opposed to rank, which I said, like I said before, the rank, i.e. the first author listed there, that's probably the one we should be working with. Each paper ID, author country, and I should be good to go, right? I should be able to map out. Except this data can get dirty. Now, for example, instead of saying China, it says PR China. It obviously stands for People's Republic of China, but that's not a standard code for us to work with, okay? And as you scroll through the data, you'll notice that there are mistakes in spelling, okay? So as you're doing your NLP analysis to try to find, because like I said, the Core 19 dataset has a bunch of tasks, and they're actually trying to figure out what to do with it. Yeah, it's funky as heck. So if you actually look at the data set, you'll notice that if I break it down, I actually have lots of things like a country of Spain, USA. Okay. So to finish off and then switch over to Chang'an, what I did basically, here's a funny one. Like there's actually literally a country called USA that literally was inside the paper. Okay. So what I did is basically I went ahead and said, let me just go ahead and get the mappings. And I actually created a map manually, which I will have given you. It's actually at the bottom of this. Okay, so this literally this map here. It's boring as heck, but basically all it is a mapping of here's the author country for each one of these values. There's a 2-digit alpha 2 and alpha 3, and so I literally listed it out and just did this manually. Fortunately, there are only 260 countries, so it wasn't that big of a deal. But the point is, long story short, there's a lot of dirty data inside there, even on what has been cleansed and organized really well. But now that I've organized it, I can map it out nicely. And as you can probably guess, the vast majority of papers for this commercial subset came from China or from the US. Okay, not that surprising, but still good to know. There's also papers from Germany and France as well. Okay, so now that I've shown you the complexities of just trying to make sense of the data and how you have to cleanse it, let's switch over to Chenyang to go ahead and talk a little bit about how we can do some cool NLP, which is the main thing about the Core 19 data set.
Thanks, Denny, for the great introduction about the data sets. My name is Chengyin Eng. You can call me Chengyin. I'm currently a data science consultant at Databricks. I work with customers by delivering data science trainings and also professional services project where I help implement data science solutions. I'm currently based in Chicago, but I did most of my undergrad or master's years in Massachusetts. I did my master's computer science at UMass Amherst, and my undergrad was in statistics and environmental studies at Mount Holyoke College. I'm going to go ahead and share my screen. It is. Okay. So as you can see here, the very top part of reading a data set is identical to what Denny has shown before. I have a bunch of data Parquet files over here, and then I'm going to read in the data in the Parquet format. And as you already know, the commercial use subset has 9,000 files, and the non-commercial use subset has just under 2,000, and then the bio sieve has under 900. And for this purpose of this NLP methods of analysis, I'm going to be using the commercial use subset data. Just to show you how it looks like if I don't do any of the cleaning, it looks like this, and it's pretty messy. You can see the abstract, back matter, beep entries, metadata. Something fortunate about this data set is that even though it's not perfectly clean, I can access a lot of information from metadata column. And if you recall from what Denny has shown before, you can see that this is a schema. So it's really just a nested JSON file. So when people think about NLP, the first thing that comes to most people's mind is, let's do some deep learning. But I'm going to show you two methods. One is using deep learning, one is not. And I'm going to start with non-deep learning method. So we're going to try to generate a word cloud from all the titles of the papers that are submitted to the organizations. And first, you need the word cloud library to be installed. So let's go ahead and take a look at what the metadata title looks like. You can see that here are the 3 examples of them. And here I'm just writing a really simple function to draw a word cloud. You can see that I'm importing word cloud and stop words from this library, and also using matplotlib as well. And here I'm just splitting all the sentences into individual words, and I'm going to pass in this clean words into this function right here. what you can see here, the argument for stop word here is what it's really doing under the hood is just removing all the common stop words. So for example, like is, are, of, and, so those words will be removed. And what is nice about this generate method is that it actually calls the function generate from frequency under the hood. What this means is that the size of the word that you will see later in the word cloud actually correlates with the frequency of the word that you see in all the titles. So you can see here, I'm just embedding the matplotlib function over here. So here I'm going to use two functions from the PyPark SQL functions called concat and also collect list. What I'm doing here is that I'm going to concatenate all the titles available in the data set. So for example, there are 9,000 rows, 9,000 different papers over here in this data set. Rather than reading them one by one, I'm going to concatenate them into all together. So then I can just pass in this entire string to my word cloud function over here. And I'm going to separate them by comma. So you can see here that I'm creating a new data frame over here, and I'm aggregating all the titles possible. I'm going to create a new column called all titles. So now I can just pass in my all titles, the string, into this word cloud. As you can see here, probably not very surprising for you, is the word infection, protein, virus, cell, human shows up as the top words in all the titles. But what we can do later is that we can remove some of the non-meaningful words from the word cloud. For example, here, we don't really know too much about using. We don't know too much about based or even viruses. We already know that coronavirus is a virus. So let's go ahead and remove them. So here, I'm just going to do some really minor modification to the function I've already written here. And I'm going to just update the stop word set. So here I'm removing the words using, base, analysis, study, research, viruses. And let's see what it looks like. So now you can see that there's no more words, there's no more using, there's no more base, there's no more viruses, but there's still virus. But so here you can see that this is the overall picture of all the titles that are available in the dataset. So as you can see here, I haven't really done much cleaning, but I was already able to do some really quick and dirty visualization about the data set that you have. So you don't even need to know deep learning to even start doing something with the data set, even though it's primarily text. So I'm going to show you now a deep learning method, which is to generate summaries from abstracts. So we know that there are 9,000 papers, and I say that I really don't have time or I'm lazy to read through all the papers. I just really want to know what is the summary, what is the important points about each paper. So I'm going to generate a summary from each abstract so that the words that you have to read is even less. So here, I'm going to use a summarizer model that's trained on BERT that initially was used to summarize lectures. There's a link over here that links to the original paper that published this model. But what it does under the hood is that it utilizes the BERT model for text embeddings and also k-means clustering to identify sentences that are closest together to generate a summary. And to use this library, you just need to install BERT Extractive Summarizer. And if you're using Databricks, you will need to install it using PyPy. So here I'm doing a really simple import over here, summarizer model. And I'm going to just take the first abstract in a data set and convert that to string. So you can see that this is the abstract that you're reading here in the first paper. Just by taking a really quick glance, this is probably about 10 rows over here. And just to show you a longer abstract, this is a second example of a longer abstract. This is probably like 20 rows. You can even scroll down even more. So this is a really long abstract. So I'm going to train a summarizer model first using minimum length parameter. And what minimum length parameter does is that whatever number you specify here, you remove any sentences that has fewer than 20 characters. So you can see the codes here are really simple and are really concise. Just one line of calling summarizer, and I define it as a model. And then I'm just passing in my abstract into this model function, model object, and I'm specifying the minimum length to be 20. So I'm going to generate the first abstract summary. And you can see here that this is now 2 sentences compared to maybe 10 sentences over here. And I'm going to look at abstract 2. can see here that rather than needing to like click into the cell and scroll down, you can see that this is significantly shorter. There's another parameter that you can use for this model, which is the maximum length. What this means is that you will remove any sentences that has more than 250 characters. So let's take a look at the first example again. If you recall, this is what we do. This is the original abstract. And I'm going to go down and take a look at the minimum length. You can see that it's now 2 sentences. But when I specify the maximum length, I would suspect that the summary would get even shorter because now all the longer sentences in the abstract is already removed. But if I generate this again, you can see that it actually is also about 2 sentences as well. So what this means is that, well, the first abstract is not that long to begin with. So playing around the minimum length parameter actually doesn't make that much of a difference. But let's take a look at the longer abstract. So here, just to call here, you can see that the number of characters and maximum length is actually the same for both cases. And here you can see that it's actually a tiny bit shorter than the one that you can see above. So hopefully this provides you showing you an example of what you can do with NLP. You don't really have to know a ton about NLP or even data science to start playing around with this data set. So hopefully that this will empower you to do something even more complicated, or even just help you to feel good about what you can do with data science as well. That's all from me.
Cool. Thanks very much. So for all you folks who actually are wanting to do the COVID-19 Open Research Dataset Challenge, this excellent setup notebook from Jinying actually should help you kickstart your NLP process. Lots of really cool little examples there that it's not going to tell you how to do those exact tasks, obviously, but at least should give you a pretty good idea of how it works. Okay, so Jinying, thanks very much. Let's finish off. Last but certainly not the least, Dhruv, please go ahead and showcase your stuff.
Thanks, Denny. Hey guys, I am Dhruv Kumar. I'm a senior solutions architect at Databricks. I've been with the company for 2 years, and before that, I've worked in a variety of big data roles. So I'm very grateful to be here and happy to speak to the intelligent audience here, because there is a pandemic up on our hands, and I feel like we as skilled practitioners of this field, we can contribute a lot back to the research and also help each other out. So as part of that initiative, what we have done at Databricks has taken some of these open data sets and put them into our repository so that you guys can start analyzing and inferring some good research from it, hopefully. Now, my goal today would be to show you A, where these data sets are located, B, how to get started with them, and C, some of the analyses I have done so far. Please, spoiler alert, there's not much going on. I've just created the data sets and come up with very, very few basic visualizations. But the hope is that this motivates you to go back and do your own experiments. In the spirit of keeping everything community research based, most of the stuff here is around open source tools, iPython, Jupyter, etc. So I'm going to be talking about how you can work in Databricks environment also with just open source techniques. So you can download this iPython notebook and work on your own systems as well. With that said, what we have done, and thanks to Denny Lee and our legal counsel, what we did was Johns Hopkins University has been publishing an aggregated data set on the coronavirus outbreak on GitHub. And this is the link for it. And in there, they go and refresh it every day. And it's a nicely formatted data set, but it has some problems. We'll come to it in a minute. But what we've done is we've taken this data set and put that into Databricks Community Edition. So there's a Databricks/datasets folder in which all this data is located. Okay. Now, because it's located over there, you can go and easily start analyzing it. You can also download this data set on your laptop and run Jupyter notebooks and start mining it for sure. But where I feel like platforms like open source cloud platforms can help is because the ability for these platforms to give you flexibility in downloading multiple data sets into one area, right? So while Johns Hopkins data set is great, it only gives you it only takes you so far. To get to some more interesting analyses, we can look at some other public data sets and also bring them into this cloud repository. So I have some research ideas, if people want to collaborate at the end of this presentation, Denny will be in touch. So what are we trying to do here? Well, what does the outbreak look like on a global scale? Let's play with some data. Let's see how far we go. Okay, so just quickly, we have, as I said, we have already loaded into Databricks datasets folder. Let's see what that folder looks like. So right now, this is the Databricks notebook. It's connected to a cluster, a machine learning cluster, which we have created. Now, if I just do %fsls, this is, by the way, a Databricks specific magic command. But if I do this, if I do Control Enter, you see I have all those other folders here, which are just a mirror copy of whatever is going on in GitHub. Okay? Now, let's look at some worldwide statistics. So we're going to try to find out at a global level, how does the epidemic look like? Okay? So because we had downloaded this data set on 17th March. That's the most latest we have so far, but so we will use that as our reference date, okay? So over here, I'm just trying to get to this particular file, on 31720.csv. You see all those files inside this GitHub repository. They are in CSV format, and the most recent ones are 17 and 18. Okay, so this is what I'm doing because it's on March 17th. Okay, so if I do that, I create that file path, and now I can just create a Spark data frame out of it. For folks who have been using Spark, this must feel very familiar. It's just a CSV. We can read its header to infer schema, and let's just see what comes up. So right now it's running Spark jobs. So you can see this is a PySpark data frame, right? So this is what, because we are still in the Spark land, so now PySpark is an extension of, PySpark data frames are an extension of just regular data frames with other bells and whistles. But as a data scientist, you can just interoperate between the two. There are also other bindings like koalas, which allow you to go back and forth between these two environments. For simplicity, what I'll do is I will just, I look at this data set. When I first downloaded it, I was like, hey, this slash here does not look very good. Like, who names the columns with slashes, right? Because it's very difficult to handle these characters later on. So first thing we're going to do is remove these columns and rename them as country and state. Okay? So let's go ahead and do that. Okay, great. Now we have state and country. Okay, so if this seems very trivial, I'm keeping it simple so that you guys can understand how this journey goes. And for folks who are not so familiar with Spark or data science programming, my motivation is it's not that difficult. You can also actually start doing some cool analyses. Okay, so now I have renamed this state and country columns. Now finally, what I'm going to do is I have two options right now. I could just continue going down the PySpark data frames path, or I can choose to convert to Pandas as well. So I tried both approaches, and there's another notebook which I have, which goes all the way into Spark. But I thought for this particular presentation, I will be using Pandas so that you guys can interoperate with other tools as well. So we can just quickly convert this PySpark data frame to Pandas using two Pandas API. And then let's see what happens when we do that. Okay. All right, great. So now if you see, this must look very familiar to folks who are using iPython Jupyter because it's formatted like that, right? Now, you must also notice one thing, that the data set, although I was able to change the country and state column names, there's still some weirdness in the state column, you see. Why is this particular row's state in Italy giving me a null value? So this is a classic big data problem where you're trying to clean and massage this data set. 80% of the problems, or rather 80% of the blockage to doing some cool analysis on data sets is not ML. It's basically data engineering and ETL and cleaning that whole thing. So this is what we are doing right now. Now, how do you handle these? Well, there's a simple path, and you can actually just go to OpenK geocode, there's a geocode API services available where you can just pass in the latitude and longitude and it'll return the state. So I tried that and it was giving me the right values, but to keep things simple, I'll omit that step right now. Now, okay, we have this data set now in latitude and longitude. How do we go about plotting it? Well, there are a bunch of different ways. One way is that we can convert these country names, China, Italy, Iran, to something called an ISO 80633 spec. There is a spec which means that country name, you convert it to 3-letter code, and then you can pass it into Databricks environment, and then we have a map, a nice looking map which comes out. This is what other presenters were showing, Denny Lee was showing you as well. Well, there's also another way. You can use Plotly. Plotly is available open source. You can just download it and install it on Databricks clusters. So that way, I have already done this. So if I look at my cluster, okay, if I look at the libraries which I've installed Plotly, Plotly-GL, and I was also hacking with Kepler-GL, which is a Uber's open source map library the other day. But the point is, you can easily install any new libraries on these clusters and start working with them. Okay, so going back to our workspace. Okay, so all right, so we were at so Plotly. Let's try to plot this guy. So because we already have latitude and longitude information, it's fairly simple and straightforward to plot. All we're going to do is pass it into this call for Mapbox, and we're gonna ask Plotly to give us a map and give us hovering data sets around confirmed and deaths. Okay, so let's see what happens. There you go. Okay. So what this is doing is essentially telling me, it has taken that same data frame, the Pandas data frame. Plotly Express was able to plot this. Mind you, the same thing can be done in Databricks as well, but because we want to be pushing these out to iPython notebooks, so it's another approach to doing that. You see, Hubei, as predicted, it had as we expected. This was just really tragic that they had 67,800. Around 67,800 confirmed cases and 3,111 deaths. If we zoom in to what's happening in United States, in our neighborhood, let's see. It's not giving me. Yeah, there we go. So mind you, this is still aggregated at the state level, but we can go further, drill down into the county level as well. And that you can do using the geocoder lookup API. It gives you not only the state, but also the county and the city name. So anyway, just some of the things you can do with data sets, showing you how to go about finding APIs online and using them in the same workflow. Lastly, once we're done with that, what else can we do? Well, Johns Hopkins is also giving us a time series data in which they are telling us every day how has the disease been progressing. So you can also, all those data sets are also located in our community editions, open data folders. And I've shown you an example of how to open it. So we can just, again, we can quickly read it through Spark and then convert to data frame and look at how the disease has been progressing per country over time. And using Plotly or Databricks internal visualizations, we can come up with some analyses and understand. Now, this is all mechanics. What are we trying to accomplish? What can we do here? One of the things is that we're trying to find out if weather can make a difference in the disease transmission. We're not sure yet. None of us are experts, and correlation is not causation. Let's not confuse with that. But just for your own visual analytics purposes to understand and donate back, you can actually download NCDC's data set. It's a temperature data set, and then relate it with these data sets here and see how those trends look like. Another one, which I was thinking last night, was that, hey, we are right now isolating ourselves, from others, staying 6 feet away, washing hands, as Denny mentions. How effective is that, number one? And number two, how effective is it whether it's actually happening or not. First of all, we have to understand that. Secondly, if it is happening, how has it been effective in containing the disease spread? So one could actually use Caltrans traffic data. It's published freely by the Department of Caltrans in Bay Area. And if you go to this link, you can subscribe and register, and you can come up with these files, which are basically 5-minute interval files on the traffic information. So essentially, use proxy of highway data as a measure of social isolation and see what it's coming, how it's being correlated with the disease transmission. Again, this goes back to our first point. While Johns Hopkins dataset gives us a lot of rich info for sure, but the magic happens when you start combining other data sets like the weather data or this DOT data, transportation data, to come up with more richer analytics. So that was a short and sweet. We're going to be refining this notebook and publishing later on today, and via Denny Lee and other channels. And back to you, Denny.
Perfect. Thanks very much. I want to be cognizant of the time right now. So since right now is a little after the hour, so we ran a little bit long. I'm sure it's my fault, so no worries, everybody. Dhruv, that was a wonderful session, very helpful. So to everybody on this session, want to remind, we will be putting these notebooks online at the Spark Global Online Meetup, as well as Seattle Spark and AI Meetup, as well as the YouTube channel. So that way the links to the notebooks, we'll probably publish the Databricks notebooks first, just because we have them already done. But by the same token, we are going to be publishing the iPython notebooks, so you can run them locally on your own environment. Hopefully this gives you a good starting point to be able to go ahead and make sense of the data, just like Dhruv called out. You can go ahead and join other data sets together to make things really interesting, just like Changing called out. There's some amazing NLP that you can do against the data sets, especially the papers there. Just like Vinny called out, there's some amazing visualizations, amazing data on whether it's the South Korean data set that she was working with or any of the other data sets. But go ahead and give things a try. See what you can get out of that data, because you can probably find some pretty interesting things. And as you do these challenges, don't just work with the data sets you've got. See, as noted, see if you can join some data sets together to find some interesting patterns. And that's it for us. I did want to finish it saying, like I said before, hopefully you guys stay safe, shelter at home, wash your hands, and do social distancing. Otherwise than that, I thank you very much. Karen, is there anything else that we need to do to close this off?
No, I think we're all